Welcome to our Introductory Biology Microlearning channel. In this video, we'll explore the fundamental principles that govern living organisms. I'm excited to dive into the fascinating world of biology with all of you. The different levels of biological complexity, from atoms and molecules to cells, tissues, organs, and entire organisms, provide the foundation for understanding life. Studying cells is crucial because they are the basic units of life, performing the essential functions necessary for an organism to survive. Understanding the characteristics of living organisms, such as their ability to grow, reproduce, respond to stimuli, and maintain homeostasis, is essential for grasping the nature of life. Exploring the principles of evolution, including how species adapt and change over time, sheds light on the origins and diversity of life on Earth. Cells are the fundamental units of life, coming in a vast array of shapes, sizes, and functions. From tiny bacterial cells to massive nerve cells, the diversity of cell types is a testament to evolutionary adaptations. Cells have evolved over billions of years, becoming increasingly specialized and efficient. The three main domains of life, archaea, bacteria, and eukarya, each represent distinct evolutionary lineages with unique cellular features. Archaea and bacteria are known as prokaryotes, with simpler cellular structures, while eukarya encompasses more complex, membrane-bound organisms. The eukarya domain is further divided into four distinct kingdoms: Animalia, Plantae, Fungi, and Protista, each with their own unique cellular characteristics. Cells are the fundamental units of life, the building blocks that make up all living organisms. They come in a wide variety of shapes, sizes, and levels of complexity, but share a common set of basic structures and functions. Cells are responsible for carrying out the essential processes that sustain life. This microscopic image shows the incredible diversity of cell types that exist in nature. Understanding the basic biology of cells is crucial for fields like medicine, biology, and biotechnology. Bacteria are single-celled microorganisms with a very simple cellular structure compared to more complex eukaryotic cells. While bacteria lack a true nucleus and membrane-bound organelles, their cellular machinery is highly efficient and sophisticated, allowing them to adapt to diverse environments. The apparent simplicity of bacterial cells is deceiving. They possess a remarkable level of complexity that enables their success as some of the most ubiquitous and adaptable life forms on the planet. This slide provides an overview of the key features that distinguish bacterial cells from the more complex eukaryotic cells found in plants, animals, and other multicellular organisms. Cell sizes vary tremendously, from the smallest known cells, just nanometers wide, to the largest cells like the ostrich egg, which can reach 15 centimeters in diameter. This huge range in cell sizes reflects the diverse functions and specializations of different cell types across the living world. The ability of cells to come in such a wide variety of sizes is a testament to the incredible adaptability and versatility of these fundamental units of life. Understanding the factors that allow for this massive variation in cell size is an important area of biological research. Studying the mechanisms that regulate cell size can provide insights into how cells function and how they are able to evolve to fulfill such a wide range of roles. Cell size is one of the most remarkable aspects of cells. They can range from microscopic bacteria to massive nerve cells in the human body. This wide spectrum of cell dimensions reflects the adaptability and specialization of these fundamental building blocks of life. The largest known cell is the ostrich egg, which can reach up to 15 centimeters in diameter. Most eukaryotic cells typically fall within the range of 10 to 100 micrometers. Prokaryotic cells, like bacteria, are generally much smaller, often 1 to 10 micrometers in length. Understanding this astounding variation in cell size is crucial for comprehending the incredible diversity of life on our planet. Prokaryotic cells, like bacteria, have a simple cellular structure without a true nucleus or membrane-bound organelles. Their genetic material is found in a single circular chromosome within the cytoplasm. Prokaryotic cells come in a variety of shapes, including spherical, rod-shaped, and spiral. They exhibit a wide range of sizes, from 0.2 to 10 micrometers in diameter. Prokaryotic cells are found in a vast array of environments, from the deepest oceans to the hottest volcanic vents. They are the most abundant and diverse organisms on Earth, making up the majority of life. Bacteria are incredibly diverse organisms that can survive in almost any environment on Earth. From extremophiles that thrive in harsh conditions to E. coli in the human gut, bacteria exhibit a vast variety of shapes, sizes, and metabolic capabilities. This diversity is a testament to the evolutionary success of bacteria, which have existed for billions of years and adapted to countless ecological niches. Understanding bacterial diversity is crucial for fields like medicine, biotechnology, and environmental science, as it allows us to harness their unique properties and behaviors. The image on this slide provides a visual representation of the incredible variety found within the bacterial world. The advances in RNA sequencing and analysis have given us important insights into the evolutionary relationships between the three major domains of life: archaea, bacteria, and eukarya. By examining the similarities and differences in ribosomal RNA, rRNA, sequences, we can infer the phylogenetic connections between these fundamental groups of organisms. The image illustrates the broad division between the prokaryotic domains, archaea and bacteria, and the eukaryotic domain, eukarya. This separation reflects the fundamental differences in cellular organization and genetic complexity between these two major branches of the tree of life. I'll highlight some of the key evolutionary insights that RNA analysis has provided about the relationships between these domains. The origin of eukaryotic cells is a fascinating topic, and the endosymbiotic theory is the leading hypothesis to explain it. According to this theory, ancient prokaryotic cells engulfed other prokaryotes, which then evolved to become organelles like mitochondria and chloroplasts within the eukaryotic cell. There is strong evidence to support this theory, including the similarities between organelles like mitochondria and chloroplasts and free-living bacteria, as well as their ability to replicate independently. This suggests that these organelles were once independent organisms that were incorporated into the eukaryotic cell over evolutionary time. Understanding the endosymbiotic origins of eukaryotic cells is crucial for comprehending the evolution of complex life on our planet. Eukaryotic cells, which have a nucleus and other membrane-bound organelles, likely evolved from simpler prokaryotic cells over billions of years. This is supported by fossil records showing the gradual increase in cell complexity over time, with the earliest cells resembling modern bacteria. Genetic analysis also reveals striking similarities between eukaryotic and certain bacterial genomes, indicating a shared evolutionary lineage. Many eukaryotic organelles like mitochondria and chloroplasts have their own DNA and resemble prokaryotic cells, suggesting they may have originated from endosymbiotic relationships between early eukaryotes and bacteria. The ability of these organelles to replicate independently within the cell further supports this endosymbiotic theory. Eukarya, or eukaryotes, represent the most complex and diverse domain of life. These organisms include all plants, animals, fungi, and many single-celled creatures. Eukaryotic cells have a defined nucleus and membrane-bound organelles, making them significantly larger and more structurally complex than prokaryotic cells. This cellular complexity allows eukaryotes to carry out a wide range of specialized functions and adaptations. Eukaryotes can thrive in virtually every ecosystem on Earth due to their advanced cellular capabilities. Viruses are an intriguing topic in biology. They exist in a gray area between living and non-living things. Viruses are not considered living organisms. They lack the characteristics of life, like independent growth and reproduction. Viruses are essentially packages of genetic material, DNA or RNA, surrounded by a protein coat. Viruses can only replicate by infecting and using the living cells of other organisms as hosts. This means viruses are completely dependent on their hosts to make more copies of themselves. Viruses are microscopic infectious agents that consist of a core of genetic material, DNA or RNA, surrounded by a protective protein coat called a capsid. The capsid can have various shapes, from simple helical or icosahedral forms to more complex structures. Viruses come in a wide range of sizes, from the tiny poliovirus, 30 nanometers, to the giant mimivirus, 750 nanometers. This variation in size reflects the diversity of viral structures and the types of genetic material they contain. Viruses come in a wide range of sizes, from the tiny poliovirus at just 30 nanometers, nm, to the massive virus at 400 nanometers. To put this into perspective, the average human cell is around 10,000 nanometers in diameter, over 300 times larger than the poliovirus. The size of a virus is an important factor in determining its structure and method of infection. Smaller viruses often have simpler shapes, while larger ones can be more complex. Understanding the scale of these microscopic entities helps us better comprehend the diversity of the viral world. The concept of what qualifies as living is a complex philosophical and scientific question. There is no single, agreed-upon definition of life, as the characteristics of living organisms can vary greatly. Many biologists consider the presence of a cell, with its ability to perform basic life functions like respiration, growth, and reproduction, as a key indicator of something being living. Certain entities like viruses, which exhibit some but not all characteristics of living things, can blur the lines and challenge our understanding of what it means to be alive. Rather than a binary classification, the concept of living may be better viewed as a spectrum, with varying degrees of complexity and the ability to carry out life processes. Cells are the fundamental building blocks of all living things, both plants and animals. They represent the smallest units capable of sustaining life, with a wide range of sizes and structures, depending on their function. Cells carry out essential processes like metabolism, growth, and reproduction. The smallest known cells are viruses, which exist on the border between living and non-living. The smallest free-living organisms are mycoplasma bacteria, which measure just 0.2 to 0.3 micrometers in diameter. In contrast, the largest known cells are the eggs of some sharks and rays, which can reach several inches in diameter. Cells are the fundamental units of life, and studying them is crucial to understanding biological processes. We employ a variety of techniques and tools to investigate the intricate structures and behaviors of cells. Microscopy, from basic light microscopes to advanced electron microscopes, allows us to observe the microscopic world within cells in great detail. Biochemical analyses, such as spectrophotometry and chromatography, enable us to identify and quantify the chemical components within cells. By combining these observational and analytical approaches, we can unravel the mysteries of cellular structure, function, and evolution. Understanding cells is essential for advancing our knowledge in fields like biology, medicine, and biotechnology. Biological membranes are essential structures that maintain the integrity of cells and organelles. They act as selective barriers, allowing the passage of specific molecules while restricting the movement of others. This compartmentalization is crucial for the proper functioning of cellular processes. Membranes not only keep cellular components together, but also maintain distinct environments within the cell. This selective permeability allows for the efficient regulation of chemical gradients, concentration differences, and signaling pathways. These mechanisms are fundamental for cellular function and communication. Cell membranes are dynamic structures that allow for the selective passage of molecules in and out of the cell. Passive diffusion is the movement of molecules down their concentration gradient, with no energy input required. This process continues until chemical equilibrium is reached. The image shows a red blood cell surrounded by the plasma, illustrating this concept of passive diffusion across the cell membrane. Molecules will continue to move in and out of the cell until the electrochemical equilibrium is achieved, maintaining the necessary balance for proper cell function. Biological membranes are selectively permeable, allowing some molecules to pass through more easily than others. This selective permeability is essential for regulating the cellular environment and controlling the movement of substances in and out of the cell. The relative permeability of the membrane to different molecules can vary greatly, depending on factors like the size, charge, and polarity of the molecules. This selective permeability is a crucial property that enables cells to maintain the appropriate concentrations of ions, nutrients, and other important substances. Understanding the permeability of biological membranes is key to understanding how cells function and how they interact with their environment. Selective channel proteins are embedded in the cell membrane and act as gatekeepers, allowing certain molecules to rapidly diffuse across the membrane through facilitated diffusion. These specialized proteins have openings or pores that only permit the passage of specific substances, such as ions or small uncharged molecules. The channel proteins provide a low-resistance pathway for these selected molecules, enabling them to move down their concentration gradient much faster than through simple diffusion alone. This facilitated diffusion process is crucial for many cellular functions, such as nerve impulse transmission and maintaining proper ion balances within the cell. Selective carrier proteins play a crucial role in facilitated diffusion, acting as molecular gates to selectively transport specific substances across the cell membrane. These proteins undergo conformational changes to allow only certain molecules to pass through, while blocking others. This selective transport mechanism is essential for maintaining the proper balance and function of the cell. Understanding how these carrier proteins work is key to understanding many important biological processes. Diffusion is the movement of molecules from high to low concentration. Passive diffusion is when molecules freely move across the cell membrane down their concentration gradient, without requiring energy from the cell. Facilitated diffusion uses specialized transport proteins in the membrane to selectively allow certain molecules to cross, even against their concentration gradient. Facilitated diffusion is more efficient than passive diffusion. The transport proteins can increase the rate of movement for specific molecules the cell needs to import or export. This allows the cell to maintain optimal concentrations of crucial substances like oxygen, glucose, and ions, without expending energy on active transport. Active transport is a crucial process that moves molecules against their concentration gradient, from an area of lower concentration to an area of higher concentration. This process requires energy input, either directly or indirectly. Primary active transport uses ADP energy to directly power the movement of molecules across the cell membrane. The sodium-potassium pump is a good example of this in animal cells. Secondary active transport, on the other hand, uses the energy stored in electrochemical gradients created by primary active transport to power the movement of other molecules. This is known as coupled transport. The key point is that active transport requires an energy input, whether it's directly from ADP or indirectly from electrochemical gradients. The active transport of sodium and potassium ions is crucial for many important cellular processes like nerve impulse transmission and muscle contraction. Cells use the Na+/K+ ADPase pump to actively pump 3 sodium ions out of the cell and 2 potassium ions into the cell. This establishes an electrochemical gradient that drives other important transport processes in the cell. The energy for this pumping comes from the hydrolysis of ADP molecules. Maintaining these concentration gradients is essential for the proper functioning of the cell. Transport across cell membranes is a critical process for maintaining proper cell function. There are 3 main types of membrane transport. Passive diffusion. Small, uncharged molecules like oxygen and carbon dioxide move down their concentration gradient without using energy. Facilitated diffusion. Larger or charged molecules use transport proteins to move down their concentration gradient without requiring energy, like ions and glucose. Active transport. Molecules are pumped against their concentration gradient using energy, usually ADP, to maintain the right balance of ions like sodium and potassium inside the cell. These different transport mechanisms allow cells to carefully control what goes in and out, which is essential for their survival and proper functioning. The nucleus is the control center of the cell, containing the genetic material and directing all cellular activities. The endoplasmic reticulum is a network of tubes and sacs that transport materials and synthesize proteins within the cell. Golgi bodies modify, sort, and package proteins and other cellular products for distribution to their destinations. Mitochondria are the powerhouses of the cell, converting nutrients into energy that the cell can use to fuel its activities. Cells are the basic building blocks of all living organisms. Cells contain specialized structures called organelles that work together to keep the cell alive and functioning. The nucleus is the control center of the cell, containing the genetic material. The endoplasmic reticulum and Golgi apparatus are responsible for protein synthesis and transport. Mitochondria are the powerhouses of the cell, producing energy through cellular respiration. The cytoskeleton provides structure and allows for movement within the cell. The nucleus is the control center of the cell, containing the genetic material critical for all cellular functions. This genetic material is organized into chromatin, a complex of DNA and associated proteins. During cell division, the chromatin condenses into discrete, rod-shaped structures known as chromosomes. Chromatin allows the lengthy DNA molecules to be packaged compactly within the nucleus. Chromosomes provide a more condensed and organized form of the genetic material during mitosis and meiosis. Understanding the distinction between chromatin and chromosomes is essential for comprehending fundamental cellular processes and inheritance. The endoplasmic reticulum, ER, is a network of interconnected membranous structures within the cell. There are two main types, the rough ER and the smooth ER. The rough ER is studded with ribosomes, which are the sites of protein synthesis. Proteins made on the rough ER are destined for export from the cell. The smooth ER lacks ribosomes and is involved in lipid synthesis, such as phospholipids and steroids. The smooth ER also regulates calcium levels within the cell. The Golgi body, also known as the Golgi apparatus, is a crucial organelle in eukaryotic cells. It's responsible for processing, sorting, and packaging proteins and lipids, acting as a central hub. The Golgi receives materials from the endoplasmic reticulum and prepares them for distribution. It modifies the proteins and lipids, adding sugars, tags, and other molecules as needed. The Golgi then packages the materials into vesicles to be transported to their final destinations, like the cell membrane, lysosomes, or other organelles. This organelle is essential for the proper functioning and organization of the cell. The Golgi body is a critical organelle in the cell that plays a key role in cargo sorting and packaging. It receives materials from the endoplasmic reticulum and acts as a central hub, sorting and packaging these materials into vesicles. These vesicles are then dispatched to their appropriate destinations within the cell or exported outside the cell. The Golgi body is essential for ensuring that cellular components and secreted materials reach their intended locations. This allows the cell to properly assemble, distribute, and export the necessary molecules and structures it needs to function. Mitochondria are often called the powerhouses of the cell. They convert chemical energy from food into a form the cell can use, called ADP. Mitochondria have their own DNA, separate from the cell's nucleus. The number of mitochondria in a cell depends on the cell's energy needs. Muscle and nerve cells have more mitochondria, are involved in other processes like calcium signaling and cell death. Mitochondria are essential for providing the energy that cells need to function. The cytoskeleton is an intricate network at the core of every cell, providing structural support and facilitating essential cellular processes. It is composed of three main components: microtubules, microfilaments, and intermediate filaments. Microtubules are the largest and most rigid, acting as cellular highways to transport materials. Microfilaments, made of actin, play a crucial role in cell motility and muscle contraction. Intermediate filaments, like keratin, confer mechanical strength and resilience to cells. This hidden framework is essential for the proper functioning and structure of cells. Microtubules are essential cytoskeletal structures found in eukaryotic cells. They provide structural support, facilitate intracellular transport, and play a crucial role in cell division. Microtubules are composed of tubulin proteins and form a dynamic network that can rapidly assemble and disassemble as needed by the cell. Their unique properties, such as polymerization and depolymerization, allow them to perform a wide range of functions. Microtubules serve as tracks for the movement of organelles, vesicles, and other cellular components, powered by motor proteins like kinesin and dynein. They also form the mitotic spindle, which is essential for chromosome separation during cell division. And there is much more to explore. Thanks for joining us in this exploration, and see you in our next video.